Okay, Bismillahirrahmanirrahim. Today's lecture is about diluting and concentrating mechanism of, and not clearance. It is diluting and concentrating mechanism of the kidney. So how the kidney will make our urine diluted and sometimes make concentrated, depending upon the water play, depending upon the water in our body. It regulates the amount of water lost in the urine. So this is one of the function. So in today's lecture, we will talk about the factors which are determining the ability of the loop of Henle to create the osmotic medullary gradient. What is mean by osmotic medullary gradient? What is gradient? Gradient means difference of different levels of osmolarity. At some place it may be 300 milliosmol per liter. At another place it may be 400. At some place it may be 500 milliosmol per liter, like that. So it will be a different levels of osmolarity will be found, and this is known as the osmotic gradient. Then the next point in the today's lecture is about what is mean by counter current multiplier. Counter current multiplier. These are the important terminologies we will be using in today's lecture. Counter current multiplier and counter current exchanger. What is counter current multiplier and what is counter current exchanger? We will see, which is required for concentrating and diluting the urine. And then the next point is about explaining the the changes in the osmolarity due to because of this counter current flow. And what is the role of ADH? What is ADH? ADH, anti-diuretic hormone. So the role of ADH is there for regulating the water balance to make a diluted urine or to make a concentrated urine, depending upon the situation. And in the last part, we will discuss about micturition reflex. Can anyone tell me what is mean by micturition? What is micturition? Also known as voiding of urine. Okay, so the bladder stores the urine for some time and it will be regularly throwing out this urine outside. This is called as micturition, and it is a reflex mechanism. Reflex mechanism. We will see what is this reflex. Okay. So urine excretion depending upon the body's state of hydration. Sometimes we are drinking lot of water. Sometimes we are drinking little amount of water, or maybe due to atmospheric condition. Like in summer or winter, different conditions, our body need of water will be changed, and accordingly the urine output will be changed. Sometimes it will be concentrated, sometimes it will be diluted. So if we drink too much of water, if we drink, let's say I drink at a time around a bottle of 2 liters of water I drink, 2 liters, what will happen? It will get added to the blood or plasma, and this will lead to decrease in the plasma osmolarity. And when it will be filtered through the kidney, then the urine will be diluted urine. The urine will be diluted, or you can say hypoosmolar. Hypoosmolar. Less osmolarity, less concentration of the solute. This is the effect of drinking a large amount of water at a time. In case of water absence or water deficit, the extracellular fluid volume will become hypertonic. Hypertonic. What is meant by hypertonic? Increase osmolarity. Above what? Above 300, because 300 is our normal osmolarity. If it is more than 300 milliosmol per liter, then we call it as hyperosmolar, hypertonic solution or hyperosmolar. So the kidney will decide depending upon the water reabsorption or water availability in the tubules. This is the main theme of today's lecture, that how the kidney will regulate to excrete a very diluted urine or a very concentrated urine. Sorry, this is not the correct picture. The drop of the urine will be small, but it will be very dark. Whereas diluted urine is a big amount, but without color. This is concentrated urine and darker in color, dark yellow color. So how this happens, we will see in today's lecture. So urine is the kidneys are capable of either throwing out less amount of water or more amount of water. It depends upon all the different regions of the nephron has got different capability of absorbing the solutes and as absorbing or reabsorbing the water. So water reabsorption is obligatory in the proximal convoluted tubule. So if you remember the parts of the nephron is has got Bowman's capsule, then proximal convoluted tubule, loop of Henle, distal convoluted tubule, and the collecting tubule and collecting duct. Okay. So in proximal convoluted tubule, 65% of the water is reabsorbed, whereas 15% of the water is reabsorbed in the loop of Henle. Whereas what is the reabsorption here in the ascending limb? Nothing. It is not permeable to water. Whereas in the distal convoluted tubule and the collecting tubule, it all depends upon the presence of ADH hormone, antidiuretic hormone, which will be responsible for absorbing according to the amount of ADH present. So final adjustment of the urine volume and the osmolarity depends upon the extent of elective. Facultative means elective. How much depends upon the how much is the concentration of ADH. Based on that, the reabsorption of water will be taking place. So level of ADH is deciding how much is absorbed from the collecting tubule. And medullary interstitium hypertonicity is also required. If you remember, the kidney has got, if you take the cut section, let's say this is a kidney. If you take a cut section, you will find that it has got two parts. Outer part is known as the cortex. Inner part is known as the medulla. So we are talking about medullary interstitium hypertonicity here. Hypertonicity in the medullary interstitium, and it will be not just one value. It will be a gradient. Different values will be there. Different levels will be found. So for concentration of the urine, suppose the person is in summer, or maybe you are traveling in a desert where you will have a limited amount of water with you, or maybe you are not drinking water. That times the kidney will try to conserve the water and not allow it to going into the urine. At that time, there will be, what are the requirements? How the kidney will do? It requires high level of ADH. ADH should be secreted, and there should be a osmolarity has to be present in the form of osmotic gradient. Different levels of osmolarity should be present in the renal medulla or medullary interstitial fluid. How the water will move whenever the water is moving from a tubule into the interstitium? What is the basic requirement? Can anyone tell me? Suppose this tubule contain water and this is a interstitium. Interstitium. How the water will move? By which method? What is the requirement? If you remember, I shown you in the first lecture that whenever we say reabsorption of water, we have to create a difference. Suppose here is this much amount of water and here is slightly bigger amount of water and it is separated by a membrane. So water will move from its higher concentration to lower concentration, or you can say from diluted area to concentrated area, right? This is the basic requirement for movement of the water. If both sides are equal, then there will be no movement of water. If we create a difference, like one place it is diluted, that means more water, and one place concentrated solution, that is less water. So water will be moving from higher to lower concentration of water. This is osmosis. This is called as osmosis. So for that, if we want to create in the medulla hyperosmotic gradient, that means water should be moved from, there should be some gradient, there should be some difference should be made between the tubular fluid and the interstitium. When we say because this is the tubular fluid is also an extracellular fluid and interstitial fluid is also extracellular fluid. If both are suppose 300 milliosmol, let's say imaginary condition that the baby is born and he is having the equal amount or normal osmolarity across all the tissues or all the cells, then it will be having 300 milliosmol inside the tubule. That is the tubular fluid, because this is the filtered fluid from the glomerulus. So same plasma, it is similar to the plasma. That's why it is 300. And also in the interstitium, let's say it is having 300 milliosmol. So will the water will move? Is there any movement of water from inside to outside or outside to inside? Is there any movement possible? Are you able to listen me? You can put your answer in the chat also. Okay. So this is the basic requirement for excreting a concentrated urine, that there should be a gradient formed in the medulla. This is the cortex, outer part, and this is the medullary part. In the medullary part, you will see the concentration is changing from 600 to 1200. That means it is creating a gradient. This is called as medullary hypertonicity or medullary concentration gradient. How it is produced, I will show you in the next few slides. So there is a gradient starting from 100 milliosmol to 1200 milliosmol per liter. Human kidney can produce osmolarity of the urine, or maximum osmolarity or maximum hypertonicity of the urine is around 1200 milliosmol. This is the capacity. We cannot increase more than that. We cannot excrete urine which is having osmolarity more than 1200 milliosmol, because this is the capacity of our kidney. Maybe if you compare human beings with some animals may have got capacity to increase concentration of the urine. That is, they can concentrate the urine to a larger extent. So what is the process by which the renal medullary interstitial fluid becomes hyperosmotic? This is the first question. Question number 1 is how this medullary interstitium, this part, medullary interstitium becomes hypertonic, increase osmolarity. How? Or how the gradient will be produced, like 400, 500, 600, 700 till 1200. Till 1200 milliosmol gradient is created. How? This is our first question. How in the kidney the gradient is created? This gradient is created by a mechanism, we call it as counter current system or mechanism. Counter current system. What is mean by counter? Counter means opposite. Bilaks. Counter means bilaks, going in opposite direction. What is mean by current? Current means flow. The fluid is flowing in this direction and the other fluid is flowing in the other direction. This is called as counter current system. We will see it in the next slides how this system works or where it is exactly located. Now, can you just go back to the first lecture where I discussed that there are two types of nephron. What are these two types of nephron? One was
cortical
okay.
and juxtamedullary.
Okay, good. So two types of nephrons were there. One was juxtamedullary, that is lying deep in their loop of Henle going deep into the medulla. Second one is the nephron which is lying in the cortex and it is not extending into the medulla. So two types of nephrons were there. One is juxtamedullary and second one is the cortical nephron. All other functions like reabsorption, secretions, or for the formation of normal urine is done by the cortical nephron. The juxtamedullary nephrons are meant for concentrating and diluting mechanism or regulating the water balance. This is the function of juxtamedullary nephron. So if you remember the picture of juxtamedullary nephron, I will show you here. They have got cortex, the glomerulus lying in the cortex, whereas the long loop of Henle is going deep into the medulla and then coming back. So there is a long loop of Henle. What is the second difference between cortical and juxtamedullary? Number is less. The juxtamedullary nephrons are less in number, whereas cortical nephrons are more in numbers. Third difference. First is long loop and deep into the medulla. Deep into medulla. Second one is numbers. Number is less in number.
The glomuli of the cortical will be in the at the outer layer of the
Okay.
cortical.
Okay. What
And
about blood? Yes. What about blood supply?
It will be for the cortical more. 90%.
Blood supply is 90% for the cortical and less for the juxtamedullary. And what is the blood vessels? How the blood vessels are there in cortical and juxtamedullary? In juxtamedullary nephron, the peritubular capillaries are known as straight vessels. Yes, vasa recta. Straight vessels. So they will be following here. Straight. Same like a loop of Henle. straight vessel. They are straight going down. Whereas in case of cortical nephrons, they are here like this, intermingling, not straight. Okay. These are peritubular capillaries. Okay. So these are the difference between the two system or two types of nephron. One is juxtamedullary are less in number. They have got long loop of Henle going deep into the medulla, and their peritubular capillaries are having vasa recti or straight vessels, and their blood flow is minimum. If you go deep into the vasa recti, it may be having only 1% of the blood flow is there, only 1%. Okay, so less blood. So that's why it will be flowing very slowly going down and supplying the blood vessels. Okay, nutrition to the tubule. Okay, coming back to this slide, that for what is counter current system? Counter current system, as I told you, counter means opposite, current means flow. So which flow is opposite? Tubular flow. One is tubular flow. In the loop of Henle, the fluid is moving in opposite direction. That means it is moving down here in the descending limb of loop of Henle, and it is moving up in the ascending limb of loop of Henle. Both are opposite to each other. They are opposite to each other. That's why it is called as counter current. What is mean by multiplier? Multiplier, just 2 into 2 is equal to 4. This is multiplication. So same pattern, same principle is used here, but I will explain you what is mean by multiplier, counter current multiplier. But the principle is same. It is multiplying the numbers and becoming a larger number. Okay, their vasa recti preserve the gradient while providing the blood to the renal flow. The vasa recti for the juxtamedullary nephron is straight, and also it is having a loop, straight and loop like this. Sorry. It is also having a loop, and also it is going opposite direction. The blood flow is down here in this segment and up in the opposite segment. So both are opposite to each other. That's why it is called as counter current. So one counter current in the tubule. This is the tubule, that is loop of Henle. Sorry. In the tubule or loop of Henle, part of the tubule is loop of Henle. And here it is in the vasa recti. Vasa recti. The flow of blood, here it is, blood is flowing in opposite direction. Here it is the glomerular filtrate. Glomerular filtrate is moving. Filtrate is moving in opposite direction. Okay. So these are the two differences you have to remember. One is the fluid which is moving in opposite direction, and it is called as counter current multiplier. Another one is blood is moving in opposite direction, slowly moving from above till down and then down till going up. This is also opposite direction, counter current, and that's why, and it is maintaining. It is maintaining the osmolarity, osmolar gradient. This is known as counter current exchanger. For the vasa recti, it is known as counter current exchanger. I will tell you what is the meaning of exchanger. Exchange means something is exchanging, going from here and down and coming back into the blood. So that is exchanger. The collecting ducts of all the nephrons uses this gradient in conjunction with the hormone vasopressin or ADH to produce a varying concentration of the urine. So this is known as the osmotic equilibrating device. The collecting tubule, based on the availability of the ADH, they may be absorbing more water or less water. Whole thing, number one, counter current multiplier. Number two, counter current exchange. And number three, the presence of ADH and effect on the collecting tubule. All three, they are known as counter current system. So counter current system means collection of all these three elements. Got it? So before we go to before we proceed to know how the kidney produces concentrated urine, we must know what is these three elements: countercurrent multiplier, countercurrent exchanger, and what is the effect of ADH on the collecting tubule. Okay. Now, if you remember, there is a specific permeability for each ion as well as for solute as well as for water, different in different parts of the nephron, different in different parts of the nephron. Comparing to the descending and ascending limb of loop of Henle, there is a different mechanisms are there for absorption of the fluid as well as for solutes. So if you remember, descending limb is highly permeable to water. This is we are talking about loop of Henle. So if you remember, the loop of Henle is highly permeable to water descending limb in the descending limb. That means this part. Okay. Here is the glomerulus and here is the proximal convoluted tubule, and then the descending limb will come. So here water is extremely permeable. It can be reabsorbed easily. Whereas there is no sodium and water reabsorption. And there is no it is impermeable to sodium. Two things are there. Water can be easily absorbed, but sodium cannot be absorbed in the descending limb. Whereas in the ascending limb, it is active transport of sodium chloride is there here in the ascending limb. What is this channel known as? If you remember, for absorption of sodium chloride and potassium.
Sodium potassium ATPase.
No, sodium potassium 2 chloride channel. It is reabsorbing sodium ion, it is reabsorbing potassium ion, and to neutralize 2 positive ions, there are absorption of the 2 chloride ion. So it is absorbing the sodium, potassium, and 2 chloride channel in the ascending, thick ascending limb of loop of Henle. But it is not absorbing water. Here, water is not reabsorbed. Here, sodium is not reabsorbed. So you must remember these things first. Then I will tell you how the counter current system will be doing its job for concentrating or diluting the urine. Okay. There is a counter current flow produced by the close proximity. So the fluid is moving in opposite direction from down here and then up here, and they are very close to each other. They are very close to each other. Okay. This is how the system is seen. This is the glomerulus. This is the Bowman's capsule, proximal convoluted tubule, descending limb of loop of Henle, ascending limb of loop of Henle, distal convoluted tubule, and this is the collecting duct, like that. Okay. Now imagine a situation where the baby is born, just born, and all the things are normal. That means the osmolarity will be how much? When we say normal, when we say the osmolarity
280.
280, or we, for the purpose of discussion, we say 300. When we say normal osmolarity, that means it is 300 inside the cell, 300 outside the cell, 300 in the plasma, 300 in the interstitial fluid. That's why we say it is osmolarity is normal, normotonic. Okay. So fluid which is filtered, because this is a blood here coming from here, it is the same. Blood is containing the solutes which are almost 300 milliosmol. So it is filtered. It will be filtered. That's why the fluid will have osmolarity of 300. Agreed or not? The filtrate which is collected in the limb of loop of Henle or descending limb of loop of Henle is same as that of the plasma. Okay. So that's why the osmolarity is 300. Got it? And also in the interstitium, we say the osmolarity is normal. That means it is 300. So both are equal. Okay. This is how the fluid is moving. Is there any possibility of absorption of water in this case? Question. In this situation where the osmolarity is 300 here and in the interstitium is also 300, will there be a movement of water?
No,
Why? Very good
because
answer.
it will be the same. The water will
Yes.
be absorbed. It will be the same amount. It
Yes,
will be
as I told you in the beginning that if the two fluids are separated by a permeable membrane and both are having same osmolarity, that means there will be no net movement. Got it? Good. Let us assume now the the function is started just now. Just now the renal tubule started functioning. Okay. What will happen? As you know, in the thick ascending limb of loop of Henle here, thick ascending limb of loop of Henle. What is there? Which type of channels are here? Which channels are here in the thick ascending limb
the
of loop of
aqua,
Henle?
the
No,
aqua too.
no. Sodium, potassium, 2 chloride channel is there, right? It is absorbing sodium ion, chloride, potassium ion, and along with it, 2 chloride ions. So from here, suppose the solution which is coming here 300, that means 300, some osmoles are there, some sodium potassium is making the osmolarity. If it comes here and there are channels here, these channels, what will they will do? They will take sodium, they will take potassium, they will take chloride. Okay. So how will the osmolarity will remain same or increase or decrease? What will happen if they take solutes? Because this segment is impermeable to water. Water cannot go out. Only solutes are lost. What will happen to this to the fluid? Will it be same or the osmolarity will increase or osmolarity will decrease? Question. From the fluid which is 300 milliosmol, if solutes are taken out because of the sodium potassium 2 chloride channel, what will happen to the concentration? This is a very simple question.
It will decrease.
It will decrease. That's why it is shown here. It will become, instead of 300, it will become 200. Got it? 200. So it will move till the collecting tubule. Everywhere it will be absorbing because of the sodium potassium 2 chloride channel, sodium potassium 2 chloride. And it is poured into where? In the interstitium. These sodium potassium 2 chloride channel are going where? In the interstitium. Right or wrong? When I shown you this
Yes, right.
picture, yeah, when I shown you this picture, that is lumen here and sodium potassium 2 chloride channel is here and interstitium here. If it is going out, it will be poured into the interstitium, sodium, potassium, and 2 chloride. So interstitial osmolarity will become same, will remain same, increase or decrease. What will happen to the osmolarity?
Increase.
Increase because solutes are added. Previously it was 300. Now few more are added. So let us assume that it becomes 400. Let us assume that it becomes 400, 400, 400 in the interstitium. Now 400 is here. And as I told you that the water, the fluid which is coming is about 300, right? So now there is a difference. Got it? It is 400 in the interstitium and 300 in the tubule. Now there is a difference. So water will move from where to where? If there is a difference of osmolarity, 300 in the tubule and 400 in the interstitium, water will move from where to where?
From interstitium to the tubule.
Opposite. Because here it is diluted fluid. I'm asking about
Mm.
water. So it will move from, it is start reabsorption of water, right? It will start reabsorption of water, and the fluid and the cycle will go on because this is a fluid flow, counter current in opposite direction. So next time it will start with 300 because 300 is the osmolarity of the plasma. When this plasma is filtered here, it will come to the down to the tip of the medullary, the loop of Henle, tip of the loop of Henle. And as it is having hyperosmolarity in the interstitium, the water will move very fast and this will become concentrated solution. This concentrated solution when goes to the ascending limb where you have got sodium potassium 2 chloride channel, sodium potassium 2 chloride channel. Now they are receiving very concentrated, so they will also use their ability to remove the sodium and chloride and potassium very fast. So again, it will increase the, it will take the solutes out and the osmolarity will decrease. Osmolarity will decrease from 400. It will, let's say 200 osmoles are removed. So 200 osmoles will be here and it will go into the next segment. This is how it will go on every time from 350 water will move, it will become concentrated. When this concentrated solution comes here, it will move many of the solutes. So the interstitium will become hyperosmolar. But you will see that there is a change, difference. 350 here, 500 here again. This is, let's say this is the cycle number 2. This is the cycle number 1. Now next is again, next cycle, cycle number 3. Again, the fluid is 300 milliosmol, but because of the concentration here is increased maybe 400. So water will move very fast and then it will become concentrated. And this concentrated solution when enter into the ascending limb where sodium potassium pump will, they will have a very concentrated solution. So they will move many solutes out, sodium, chloride and potassium. Again, this gradient will increase and the fluid which is passing here, now it will become diluted. Okay, diluted to the collecting tubule. Same thing will be repeated. Cycle number 4, 5, till what time? Till the maximum osmolarity is created in the interstitium. And I told you the kidneys, human kidneys osmolarity maximum capacity is up to 1200 milliosmol only. So till that time, the whole process will be repeated. And if you observe, every time the fluid is moving, it is increasing the osmolarity. Previously it is 400. Here it is 400 milliosmol. In the next cycle, it becomes 500. In the next cycle, it becomes, let's say 600. In the next, here it is 600 and it is becoming here 1200. So every time fluid moves, it is increasing or multiplying the osmolarity of the interstitium. That's why this is known as counter current multiplication. 2 into 2 is equal to 4. 2 into 2 is equal to 4. That's why here multiplication of the osmolarity is happening. That's why it is called as counter current multiplier system. This is regarding the loop of Henle. This is regarding the loop of Henle of juxtamedullary nephron, not about the cortical nephron, not about the blood vessel. It is about the nephron or loop of Henle of the nephron, which is increasing the osmolarity of the medullary interstitium. That's why it is known as counter current multiplication. Counter because movement is opposite, current is flow of tubular fluid, and increasing the osmolarity is multiplication. Okay, are you getting my point? This is the whole picture. In the book, you will find a picture on the same page like this, where you will see that it is you can see all these are increasing from 300 to 500, then 500 to 600, 600 to 1200. So it is every cycle there will be increase in the osmolarity of the interstitium. That's why it is called as counter current multiplier system. What is the benefit? What is the benefit of creating this gradient? Counter current multiplication system. What purpose it will serve? It will establish a vertical osmotic gradient from cortex to the medulla. You will have osmolarity of different types. Like here, the osmolarity may be 300, then 400, then 500, 600, like this, till in the deepest medulla, it will be 1200. So it is a vertical from above downward. The osmolarity is increasing because of the arrangement of the deep or juxtamedullary nephron. Gradient means this difference, 300, 400, in turn used by the collecting ducts to concentrate the tubular fluid so that the urine will become more concentrated when the body is not allowing to excrete the more amount of the water. Second, the effect that fluid is hypotonic as it enter into the distal tubules will enable the kidneys to excrete urine more dilute than the normal fluid. And it is in absence of ADH. If the ADH is there, then this fluid will be completely reabsorbed. Okay. Role of vasopressin. We have seen role of counter current multiplier system. Now we will see role of vasopressin or ADH. Vasopressin control variable amount of absorption in the final tubular segment, that is the distal convoluted tubule and the collecting tubule. So 65% we have seen reabsorption is obligatory in the proximal tubule, PCT, and then 15% in the loop of Henle, and in the variable secretion of variable amount will be absorbed under the influence of ADH. What the vasopressin will cause? Vasopressin will increase the permeability. As somebody told me in the beginning, that it will increase the channels, aquaporine channels. Aquaporine channels will be become more, and it will cause more reabsorption of water. Vasopressin works on the tubule cells through cyclic AMP mechanism. During the water deficit, when the water is less, then vasopressin concentration will be very high in the blood. If suppose the person is fasting, or maybe he is in desert and not drinking proper amount of water, that time ADH, if you observe, if you take the sample of blood from this patient or person, you will find increased ADH secretion. Increased ADH amount will be found in case of deficiency of water. During the excess of water, whereas if the person drink, let's say there is a competition. Let's say we are making a competition in our classroom who drinks a lot amount of water. So one person drink, let's say, one bottle of this type, around 200 ml. Another person drink 200 ml, two bottles. Another person drink 5 bottles of like this. Another person drink 10 bottles of like this. And just it was a because of the competition. What is happening? We are overloading the body with excess of water. This will lead to decrease in ADH secretion. ADH will be decreased in the blood. If ADH is decreased, what will happen to the tubule? Renal tubule, collecting duct, what will happen? Please answer quickly. These are basic questions. If you drink lot of water, maybe 2-3 liters at a time, what will happen to the ADH? ADH will decrease, but what will happen to the tubular fluid or urine?
It will increase.
Diluted.
Diluted. Diluted urine will be formed. Diluted means osmolarity is how much? Could be how much? Hmm? Is it 300 or is it 600? or is it 1200? Or is it 100? What is the answer? If you drink lot of water at a time, let's say I drink 10 bottles of like this, which is 2 liters, because one is 200 ml. So if I drink one by one each, then my urine will become very dark, right? Please find the answer. So this is the mechanism of action of vasopressin. When vasopressin is released in the body, it will be going, it will mix with the blood. From where the vasopressin comes? Vasopressin coming from posterior pituitary gland. Okay. And then it will be through the blood, it will act on the collecting tubule where through via the V2 receptor. This will lead to the formation of many channels. You will find that it will make many channels inside, and these channels will be located on the luminal border for absorption of water. So water will be absorbed because of excess. Many aquaporin channels will be aligned or fixed here on the luminal border. This is the action of vasopressin. Okay, one more factor is also there in making the hyperosmolar concentration of the medulla, and that is urea. Urea recycling, recirculation or recycling. Anyone knows how the urea is absorbed? If you remember from the previous lecture, what is different regions and how much amount of urea is reabsorbed and where? I shown you in the previous class. This is a diagram of nephron. So as the fluid reaches down, because water is reabsorbed, water is freely reabsorbed, okay? And also the sodium chloride, This area becomes very concentrated and it contains urea also. So 50% of the urea will be reabsorbed here. Once it goes here, then 50% of urea will be added. So previous 50%, now 50% is added. So it will become total 100% from here. Then once it goes here, under the influence of the ADH, again 50% of the urea will be reabsorbed. So how much urea is lost? Only 50%. This is the normal circulation of the urea. So urea is coming out into the interstitium, then going into the fluid, then coming out into the interstitium. This is called as urea recirculation. Urea is passively reabsorbed in the proximal tubule in presence of ADH. Water is reabsorbed in the distal and collecting tubules, and urea concentration of urea in this part of the nephron, making it concentrated. Inner medullary collecting tubule is highly permeable to urea. That's why urea will be shift again back into the interstitium. This will increase. What it will increase? Medullary osmolarity. Medullary interstitial osmolarity is increased. So one thing is we have seen countercurrent multiplier. Second one is urea cycling, recycling. Here it is shown. Suppose 4.5 ml or percent or milligram is filtered. Out of that, because of the concentration, it will get concentrated here. It becomes 15, and then because of it, it will be reabsorbed. And then once it reaches here, again 50% will be added. Once it comes here, again it will be reabsorbed. So this is how the urea will be recycled, and maybe under the influence of ADH, 20% will be going into the urine. Sorry. This is called as urea recycling, and this will create medullary hyperosmolarity. Okay. So this slide is the final slide of how the body regulates, or how the kidney regulates the water balance. In case of water deficit, when fluid is entering first time into the tubule, it is 300 milliosmolar. When it goes down, it will becomes hyperosmolar, that is around 1200. Again, when it comes to the above, it will become hypoosmolar because the solutes are reabsorbed. Water is as it is. When it comes here again, it will be water reabsorption under the influence of ADH. This will lead to again hyperosmolar, highly concentrated urine will be passed outside. This is called as, this is the regulation of water reabsorption. And we have got role of three things. One is counter current multiplier. Multiplier means it's a loop of Henle which is multiplying the, the concentration of the fluid. And there is role of ADH. Okay. So loop of Henle is known as counter current multiplier. In case when the person is drinking lot of amount of water, still the same thing will happen, that it will be becoming hyperosmolar till it goes to the tip. When it comes above, it will become hypoosmolar. And as there is no ADH, ADH is suppressed because of the excessive water, decreased level of ADH. So it will go as it is. That means large amount of urine with little solute, hypoosmolar or diluted urine. Diluted urine will be lost. Okay. Looking at the arrangement of the blood vessels, if suppose the loop of Henle is creating, because loop of Henle is creating the hyperosmolar starting from 300, 400, 500, 600 up till 1200 osmolarity gradient. If it is like this, if you have simultaneously at the same side or along this side of the interstitium, if you have a blood vessel which is containing blood, what is the osmolarity of the normal blood? 300. Okay. So when it passes down, it is having a difference 300 and 500 or 400 and 300. What will happen? It will take the solutes from here. It will take the solutes from here. Take till it becomes 1200. And here it will become 300 or less than that. And it will go to the circulation. It will go to the circulation. So whatever our work was done by a loop of Henle will be lost. All this loop, the loop of Henle work, which is multiplication of the osmolarity, will be lost because of the vessel, because of the blood vessels, peritubular capillaries. But nature has given some arrangement, a specific arrangement for this region. First of all, the blood flow is very slow in the medulla, very slow, very slow, and less amount, only 1% of the blood is passing. So very less amount of the blood is going. So it is highly less chance that it will take the osmolarity along with it. One thing. Second is the arrangement is not like a straight vessels. Okay, it is called as a straight vessel because it is along the side of the loop of Henle, but it is also bending along the side of the loop of Henle. This is the loop of Henle in the center, and here on the sides it is the peritubular capillaries, or you can say vasa recti. Here they are called as, actually they are along the side of the tubule. That's why peritubular, but the name is given as vasa recti. Straight vessels, because straight down. So they are bent in the U-shaped. They are bent in U-shaped, like a loop, like a loop. So as you are rightly guessing that once it is 300 here, because of the hyperosmolarity, the solutes will be in and water will be out till the time the inside will become 1200, same as that of the surrounding. But once it goes up, then water will move in, because here it is water and here it is excessive solute. So solutes will be there and the water will move to dilute. Water will move to dilute. So it will become 1200, 900, 600, 300. And again, it will go back to the 300 original value. So the loop is to create, to keep the interstitium as it is. It will not allow the osmolarity to lost because of the flow of the blood. First of all, flow is very slow. So it will not be lost, or if it is lost, it is very little amount. Second thing is, again, to avoid this loss, the flow is made in terms of upside down, or you can say one side it is going down, second side it is coming up. We call it as again counter and flow. That's why counter current flow, but this is not for fluid, this is for blood. And it is known as counter, as the solutes are coming in and water is going out. In the other segment, solutes are going out and water is coming in. That is called as exchange. Exchange. So this is why the arrangement of vasa recti is known as counter current exchanger. Remember, two different terminologies. Counter current is same. Flow is in the two opposite direction. But one is exchanger, another one is multiplier. So multiplier is the loop of Henle, whereas counter current exchanger is the vasa recti. And it is just maintaining, the interstitial, medullary interstitial osmolarity, medullary interstitial osmolarity, right? So the function of vasa recti is to maintain the medullary interstitial osmolarity. Creation of medullary interstitium osmolarity is the function of counter current multiplier. So here it is shown in a clear way that solutes are moving in when the fluid, when the blood is going down. Solutes are moving in and the water is coming out. Water is coming out. Water is coming out. On the other side, when the fluid is going in the opposite direction, the water is moving in and solutes are coming out. So this is at two places exchange is taking place. This is called as exchanger, counter current exchanger. Vasa recti are serve as a counter current exchanger. Vasa recti blood flow is very slow and it is only 1 to 2% of the renal blood flow. That's why there will be no loss of osmolarity, or osmoles will not be lost because it is very slow. Just like imagine a situation where you are cleaning your corridor or backyard when there is a lot of dust. You use water pipe or hose. If you are applying a pressure to the water, you can clean the dirt. But if you don't apply the pressure, there will be no cleaning of the dirt. So here also in the vasa recti, the flow is very slow and it is looped. That's why the constant, the dirt means here is the osmoles. So osmoles will not be lost from the medullary interstitium. Okay, coming to the when the kidneys don't function, what is the function of the kidney? Kidney's function is to remove the waste products. If the kidney is not removing the waste products, then we say kidneys are failing, or we call it as renal failure. What are the causes of renal failure? Renal failure could be due to infections by the organism, or maybe because of some toxic material which is destroying the filtrating membrane, or maybe inappropriate immune response. Antigen antibody response can lead to blockage of these filtrating membrane, Obstruction to the urine flow may be renal calculi, renal calculi or stone can lead to abnormality of the function. We call it as renal failure or insufficient blood flow to the kidneys. In case of shock, when kidneys are not receiving proper amount of blood, normally it is 20% of the cardiac output goes to the kidney. 20% of cardiac output goes to the kidney. If they are receiving only 1%, suppose, then the renal function will not be proper and we say renal failure. Okay, coming to the next topic, that is the second topic of today's lecture, is micturition reflex. What is mean by micturition? If you remember the renal system, it has got kidneys, there are ureters. These ureters are opening into the bladder for storage of the urine, which is connected to the urethra, and urethra will open outside. Okay, so here the urine will be collected in the urinary bladder, but this bladder will hold the urine for some time as you are doing some work or sleeping or maybe in lecture or maybe in job or maybe drawing. So at that time the urine will be held into the bladder. When the situation is okay, you go to the bathroom and you relax the sphincter and the urine will come out. This coming out of urine is known as micturition. And this occurs by a mechanism or by a reflex mechanism through the spinal cord here. The arrangement is through the spinal cord. We call it as reflex, micturition reflex. This will lead to this. First, there will be some sensation of the urine in the bladder, goes to the spinal cord. Spinal cord will send the signal to cause the contraction of the bladder and relaxation of the sphincter to come to avoid the urine. This is called as a reflex mechanism. It is known as micturition reflex. So how it occurs, we will see in detail now. There are stretch receptors present in the muscle or wall or the smooth muscles of the bladder, and they are connected through the neurons, which are sympathetic as well as parasympathetic to the spinal cord at the level of S234. S234. So it is connected there. So in the sacral segment of the spinal cord as well as in the lumbar region, that is L2, sympathetic will be here. So these are the nerve fibers or connections there. Lumbar 2, 1 and 2, and mostly the 2. And the sacral, that is S2, S3, S4 segment, it is connected through neurons. I will show you in the picture which type of neuron, that is sympathetic and parasympathetic, two different things, as well as the somatic. So what is micturition reflex means? Micturition reflex means relaxation of the external urethral sphincter muscles, which will allow the passage of the urine out of the body through the urethra. And it is under voluntary control. That means I can control. If suppose a person is doing some work, at that time if he feels the urge to pass the urine, still he can hold it for some time, and then when he stop functioning or maybe the work is done, he will go to the bathroom and void the urine. This is, so it has got voluntary control also. It has got voluntary control also, but cannot delay indefinitely. That means you cannot hold from morning till evening. You cannot delay but for some time you can. What is urinary incontinence? It is inability to prevent the discharge of the urine. Normally, urine should not come out every time. Every second it should not come out. Whenever we try, that time it should come out. If it is coming out drop by drop anytime, this is called as incontinence. We are not able to hold the urine. Okay. Okay, this is a picture showing you the detailed things. This is the cut section of the urinary bladder, showing you that these are the two spots where the urethra is opening. Ureters are opening. Sorry, ureters are opening, and then it is connected. It will be going giving out the urine through the internal sphincter here, And there is a voluntary sphincter which is called as the external urethral sphincter. External urethral sphincter. This one is internal urethral sphincter. And from the spinal cord, that is, suppose this is a spinal cord here. There is a L1, L2 segment, and here S2, S3, S4 segment. And there are nerves which are supplying. So three types of nerves, a nerve supply is there. One is sympathetic. One is parasympathetic. And somatic. Somatic is the pudendal nerve. Pudendal nerve, which is supplying to the external urethral sphincter. External urethral sphincter under voluntary control through pudendal nerve, whereas sympathetic through hypogastric nerve. Hypogastric, and parasympathetic is through the pelvic nerve. Okay. What is the function of parasympathetic? Is to store the urine. Sympathetics are to hold the urine or store the urine. S for store, S for sympathetic. Okay. Parasympathetic for voiding the urine. You want to give out the urine, voiding the urine. And for pudendal nerve, it is for the voluntary control. Pudendal nerve is voluntary control of the urine. It is holding or controlling the external urethral sphincter. Okay, this is again the same diagram showing you that sympathetic is coming mainly from L2 segment through the hypogastric nerve, and it is supplying to the bladder wall as well as to the neck region, or maybe you can say internal urethral sphincter. Parasympathetic nerve again supplying the wall of the smooth muscles of the bladder as well as to the urethra. Here it is parasympathetic acting through the M3 receptor, muscarinic receptor, M3 receptor. Sympathetic are acting through beta 3 receptor, beta 1, beta 2, beta 3 receptor. Okay. Pudendal nerve are the somatic nerve. Okay. Also, this is not the only. This is only at the level of the lumbosacral spinal cord, or make you can say simply at the level of the spinal cord. The center is for this reflex. Micturition reflex center is at the level of the spinal cord. But there is also voluntary control. Voluntary control is also there from the brain, that is midbrain and pons. Midbrain and pontine center also has got control over the spinal cord. So it has got cerebral from the cerebral cortex to the midbrain, and from the cerebral cortex to the pons, and then to the lower down centers. So there is some higher centers also, but local reflex is through spinal cord. How it happens? This diagram shows you that when, let's say these are the kidneys, I will draw it very small here. They are both sides. Sending the drops of urine into the bladder, So if the drops are collected here, what is happening when there is a collection of about 10 to 15 ml, suppose 10 to 15 ml of the urine is collected here. Let's say first the person is micturated or clean the bladder or just gone to the bathroom and voided the urine. So there is no urine. Now after that, at that time when there is empty bladder, in case of empty bladder, the pressure will be zero, around 0 centimeter of water, or you can say millimeter, but here we are measuring the pressure in centimeter of water. So 0 centimeter of water is there. When 10 to 15 ml of the fluid is collected or urine is collected, this pressure will increase. This pressure will increase. I will draw it with a different pen. So 0 is when no fluid. Volume of urine is there on the x-axis and pressure on the y-axis. Bladder pressure. Bladder pressure is known as vesical pressure. Vesical pressure or the bladder pressure. So when there is no urine, the pressure is 0. When about 50 ml of the urine is collected, let's say this is the 50 ml, then there will be rise in the pressure of about 10 centimeter of water. Here it is written as 10 centimeter of water pressure will be measured. When you will have collection of fluid around 10 to 15 ml, there will be slight rise in the pressure will be there. Okay. When the fluid the urine get collected more and more, the pressure will start to rise. When the urine is collected in the bladder, the pressure will be start to rise slowly till 400 ml of the urine. Till 400 ml of the urine is collected. But before that, when around 150 ml of the fluid or the urine is collected somewhere here, the person will start to feel the contraction of the bladder. First time. Please note, when the fluid enters into the bladder, the pressure will slowly increase. A time will come when around 150 ml of the urine is collected in the urinary bladder. That time, for the first time, the person will start to feel the contraction of the bladder. Bladder will start to contract to void the urine. At what amount? Around 150 ml. But these contractions will not last longer. These contractions will not last longer. They will appear and disappear. When more urine is collected, again there will be slightly more contraction and relaxation, but still it will not cause the urination or the micturition reflex to occur. These are reflexes occurring. These are contraction of the bladder is occurring, but they are not long lasting. It is just coming and going, coming and going. If the situation or the facility is there for bathroom, the person will go and can pass the urine. But the first time he will start to feel the urge to pass the urine is about 150 ml. Before that, no. Before that, it will be just collected unnoticed. There may be slight change in the pressure. A point will come when the amount of urine is becomes 400 ml in the bladder. That time the contractions will be stronger, very strong. Then you must go to the to pass the urine. Go to the bathroom to pass the urine. Still, if that time also, if you hold the urine, then you will start to feel uncomfortable. The person will become uncomfortable after 400 ml. And if still he is holding the urine, the pressure will be tremendously rising and he will start to feel pain in the bladder. Got it? Okay. What is happening from the voluntary control? Why the bladder is not emptying directly? Because there is some higher center located which is causing the inhibition of these neurons, inhibition of this segment. They will have an inhibitory signal here in the sacral segment. They will not allow the reflex to occur and pass the urine. So when the bladder starts to fill, let me change the color. When the bladder starts to fill, there will be some stretch receptors will get stretched and they will send signal and it will go through the parasympathetic nerve, which will cause the contraction of the detrusor muscle of the urinary bladder. Then, if the situation is suitable, the bladder will contract and the internal sphincter will relax and there will be urination. If the person is at home or maybe in the bathroom, if the contractions becomes more, he will just relax the internal sphincter and there will be urination. But if the situation is not in the normal control, or maybe person is doing some work, that time the cerebral cortex will send signals to the or mainly from the pons micturating center. The signals will come to the motor neuron of the external sphincter, which will cause contraction of the external sphincter, and there will be closure of the external sphincter, external urethral sphincter, and there will be no urination. This is voluntary control. If opposite situation, if the situation is you can say favorable situation is there, person is not doing anything and he is in a place where he can pass the urine, then it will be going to the external urethral sphincter, which will be inhibited and it will cause urination. This is how the higher center is controlling the reflex center in the spinal cord. Okay, actually it can be done through the spinal cord reflex center. Okay, from here to the urinary bladder can happen, but there is a inhibitory signals are coming from the higher centers like the cortex and the pons, which will control this reflex to stop or start. Okay. What are the abnormalities of the micturition? Abnormalities of the micturition. Suppose there is a sensory nerve damage or the destruction of the sensory nerve, which is making aware you about the collection of urine in the bladder. If these sensory nerves get cut or destructed or maybe infected because of some reason, what will happen? There will be no sensation of micturition. There will be no sensation. The bladder, which is supposed to be here as a initial before it is emptied bladder, is like this. It will go on increasing the size, but there is no reflex mechanism. It will go on increasing because of the collection of the urine. And there will be a time will come when the pressure is very high in the bladder to cause leakage of the urine, and urine will becomes overflowing. Urine will overflow the bladder. This is called as overflow incontinence. Overflow incontinence because of the loss of sensory neuron. This sensory neuron can be due to injury to the spinal cord at the level of the sacral region. At the level of the sacral region, if the spinal cord is cut here, if it is cut, then the person will suffer from atonic bladder. There will be no contraction. It will go on increasing till a time will come when there will be overflow of the urine. Also, some diseases can lead to atonic bladder, like inflammation of these nerves, like even long-standing diabetes mellitus or neurosyphilis, which is affecting the sacral segment of the spinal cord, can lead to the atonic bladder, or some infection, neural infection, herpes infection of the nerves can lead to atonic bladder. Second one, another abnormality is known as automatic bladder. Automatic bladder is caused by the spinal cord damage above the sacral segment. Here it is okay, but the damage may be somewhere here. This is S2, S3, S4 segment. Here there is normal connection between the bladder and the neurons. There is normal connection, but there is no inhibitory control coming from the above, because above is the fibers are coming from the central nervous system, pons and the cortex. Typical micturition can occur. Here, because there is intact nerve supply, it is a local response, it is there, but it is not controlled by the brain. That's why this is usually found in case of spinal shock in the first few weeks of the spinal shock, when there is a sudden loss of facilitative impulses coming from the brain. Gradually, there will be reflex will start, and it will be periodically, it will, let's say, 200 ml of the urine or 400 ml of the urine is collected, and there will be normal reflex and passage of the urine. Patient can also initiate the impulse. Patient can also initiate the impulse by scratching the or tickling the thigh or inner side of the thigh or the area, perineal region, so that this reflex will be started and he can pass the urine. But there is no inhibitory control from the above. This is known as automatic bladder. Anytime, without any sensation, there will be reflex will start and the person will pass the urine. Third abnormality of the micturition reflex is uninhibited neurogenic bladder caused by, again, lack of inhibitory signals from the brain, which results in frequent and relatively uncontrolled micturition, or maybe from partial damage to the spinal cord or the brain stem, which will interrupt the inhibitory signal. So either partial damage, sometimes stroke, or sometimes the half of the spinal cord or the higher centers are damaged. This will lead to frequent urination without any inhibition. This is called as uninhibited neurogenic bladder. Okay, this is all about two important mechanism, or you can say concept we covered. One is how the kidney regulate, what it regulates, water balance, or you can say how it will pass concentrated and diluted urine, how the kidney will pass concentrated or diluted urine. For that, we have seen there is a mechanism called as counter current multiplier system, which consists of two things. One is counter current multiplier, which is nothing but the loop of Henle. And another one is counter current exchanger, which is nothing but the vasorectile. These two together will work along with ADH, that is antidiuretic hormone, to cause the either formation of diluted urine or concentrated urine. Second half of the lecture, we have seen how the micturition reflex is operated, what are the things, how the control mechanism is from the local, that is sacral region, as well as from the higher center. I hope you understand this lecture. If any difficulty, please let me know. Send me message, or if you didn't understand any part, you can just highlight that part and send me on WhatsApp. Thank you very much. Yes, question. If you have question, you can ask me. No question?